Hi everyone, we're Dana.
And Albiona, juniors from Francis Lewis High School.
Today we're going to present our experiment about the effect of lavender, thyme, and raspberry essential oils on Cranibacterium spp. So we will begin by going over the objectives. So the first one is to investigate the effect of the essential oils of lavender, thyme, raspberry, and their combination on the bacterial growth of Cranibacterium spp in vitro. These essential oils and their combination could potentially be used as antibacterial agents of Cranibacterium cirrhosis, which could provide a biosustainable alternative to the use of common deodorants that contain potentially harmful compounds. Next, we will move on to the introduction where we will begin by discussing the importance of the research. Research has developed hygienic products that can be used to mask unpleasant odors. Deodorants are highly advertised in the media as a remedy for these smells. These bad odors are mainly caused by gram-positive bacteria present in our bodies. Past investigations demonstrated deodorants are particularly leading causes of fragrance allergy. This could be explained by the unhealthy composition of the fragrance chemical. Other investigations that showed that aluminum compounds, which are found in deodorants, are neurotoxic, cause skin irritation, and interfere with estrogen production, increasing the incidence of breast cancer. Butane, which is also found in deodorants, is absorbed by different tissues in our bodies, causing irritation once it is in contact. Butane poisoning affects the central nervous system and the cardiovascular system. Additionally, commercial deodorants contain environmental pollutants that would affect air and water, leading to indoor and outdoor pollution. Before we discuss Cyanobacterium cirrhosis, we'll first explain the role that sweat secretion plays. The secretion of sweat by the apocrine glands within the underarm is caused by the metabolic activity of the cells. This is a mechanism in which the human body thermoregulates. Sweat is composed of lipids, sodium chloride, urea, steroids, and proteins that are digested by microorganisms. This biotransformation of these components into volatile fatty acids and thioalcohols are considered to be the unpleasant odors emitted. Corynebacterium, which is one of these microorganisms, produces lipases which break down the lipids in sweat to create butyric acid, which releases this type of odors. Corynebacterium cirrhosis is a gram-positive bacteria that can normally be found on the skin of both humans and animals. It is yellowish-brown in appearance and it's often found in dried form. It is non-hemolytic and it can ferment glucose and sucrose and other carbohydrates. It doesn't usually cause disease, however, it occasionally causes endocarditis and skin infections. Now, we will talk about the essential oils, which are treatments we will be using in our experiment. Essential oils popularity is increasing as a result of the high demand of new active ingredients from natural renewable sources that are more eco-sustainable and safe. When essential oils are inhaled, they activate the olfactory system, triggering specific physiological responses. Past studies showed several types of essential oils can inhibit bacterial growth. Within many plants, there is the presence of phenolic acids, which have antibacterial capabilities due to their redox properties. These compounds can also restrict bacterial resistance. Essential oils are classified as natural concentrated compounds extracted from aromatic plants. They are used for a variety of reasons, including medicinal use, perfumes, aromatherapy, and cooking. These oils can be derived from seeds, leaves, petals, stems, peels, roots, and fruits. A similar study in 2008 tested the effectiveness of raspberry juice on inhibiting the growth of Bacillus subtilis and Escherichia coli and showed that the juice itself can inhibit the growth of B. subtilis longer than the water and methanol extracts from the pulp, and also that the juice inhibited E. coli growth for the entire experiment. Another investigation in 2011 demonstrated that thyme essential oil has strong antimicrobial properties since it decreased the number of Staphylococcus sp strains by over 90% at extremely low concentrations. Its antiseptic properties are the result of its high content of phenolic compounds such as thymol and carvacrol. Table 1 shows our experimental setup. We had 4 experimental groups and 1 control group. The first 3 experimental groups were each tested independently with pure essential oil extracts, using the same volume for each treatment. The fourth group was treated with a combination of all the essential oils in equal proportions. The control group was treated with water.
First, we prepared our agar plates, where we began by autoclaving 625 milliliters of solid brain-heart diffusion agar, which is the growth medium that supports the growth of Corynebacterium supplement. We gathered 25 petri dishes, meaning that we have 5 groups and a repetition of 5 plates per group. We made sure to label them with corresponding letters based on the treatment that was applied on them, and we poured about 3 milliliters of the liquid agar into these plates. We made sure to wait for them to solidify, then we would flip them over to prevent condensation of falling onto the agar and causing contaminations, and they were properly stored in the fridge to prevent contamination for us to use later on. Then we will prepare our nutrient broth solution, where 2 grams of nutrient broth powder is poured into 100 milliliters of deionized water in a 300 milliliter Erlenmeyer flask. Then the solution will be autoclaved. From the Corynebacterium cirrhosis culture, we will grab a sample using a sterile loop, and we submerge the loop into the autoclaved broth solution. Then this flask will be placed in an incubator at 37 degrees Celsius to grow the bacteria. Using previously prepared nutrient broth solution and agar plates, we cultivate our Corynebacterium cirrhosis with respective treatments. Using a micropipette, we dispense 200 microliters of the broth on the agar plates. Then, using a spreader, we'll evenly spread the bacteria throughout each plate. Then we will take 4 soap discs with respective treatments, and they will be placed on the respective agar plates. We will incubate these plates at 37 degrees to grow the bacteria, and we will measure the rate of inhibition for a period of a week. Our results consist of 3 different trials and 2 figures per trial. For Figure 1, Trial 1, it measures the means of zone of inhibition per day. This is for a span of 5 days, and it was clearly seen that the lavender and raspberry oil exerted the highest inhibition of the bacterial growth. And for Figure 4, Trial 1 shows the means of zone of inhibition trends, and this was taken over 8 days, and it was seen that the highest trend was shown by raspberry and lavender oil in inhibition of the bacteria. For Trial 2, it was clearly seen that for Figure 2 and Figure 5, the overall trend showed that thyme essential oil had the greatest effect on the inhibition of Corynebacterium supplement. Unlike Trial 1 and Trial 2, for our Trial 3, Figures number 3 and number 6, it was shown that the most significant inhibitory effect was from raspberry and thyme oils. Before we go into our conclusion, I want to take into account some limitations and errors that have occurred through our experiment and could have impacted our overall data. One thing I wanted to look into was the fluctuating temperatures, since these petri dishes were taken in and out of an incubator to do measurements and to observe. These fluctuating temperatures could have caused and impacted the effect of the oils overall, and could have also impacted the growth of bacteria, as well as the contamination of oils. If these oils were contaminated, since they weren't autoclaved, since we believed they were already sterile, these oils could have been contaminated and could have not done as great as and given as great as results as they would if they were sterile. And human error occurs in every experiment and every study, and in our experiment specifically, it could have occurred when we were measuring the radius, which was done by not using the proper lab material. In our conclusion, I would like to talk about our overall results and most significant results, and also expand on how this is important for our research and experiment. Overall, throughout trial and error, the data has shown that these 3 oils and their combinations have an inhibitory effect on Corynebacterium supplement. This further expanded on our research and our hypothesis, showing that these oils have inhibited the growth of bacteria and are further reliable compared to deodorants. Even though our hypothesis stated that the combination of the oils would have the greatest results, even though that was not valid, there was still some inhibition shown and exerted from the combination of these oils. And it was suggested that thyme and raspberry essential oils had the greatest inhibitory effect in the bacterial growth of Corynebacterium cirrhosis, being the better alternative treatment. So, after taking into consideration and looking into our conclusion and also some limitations that we had, some things we could do to improve our future work is by increasing trials, going into 7 consecutive days, measuring samples more than once, and also increasing of the sample size. This could help reduce human error and give us better results and more accurate results. And for future work, we can also compare oils to normal treatment towards this bacteria, which is a deodorant, to have a better comparison on the inhibition rate and to see whether or not the bacteria growth is inhibited more by the deodorant or by the oils. To wrap up this presentation, these are all the references we have used through our experiment. Hopefully you enjoyed our presentation and you learned something new today. Thank you so much for your time.